So this last presentation will be about variant annotation. So as you have seen and as you probably know, if you do variant analysis, then you will probably find really a lot of variants. But out of these variants, you want to, of course, extract the most important variant. And typically, the most important variants are variants that have an effect on, for example, the amino acid sequence of a protein, because that later on can have an effect on the phenotype. So when you do that, then you are doing something that is called functional annotation. So that would be variants that are relative to genomic features, and often those genomic features are genes. So variants with big effects on those genes are either in coding regions or in promoter regions, because those kind of regions are important for the protein. Less important are, for example, variants in intronic regions, because they will not be translated into amino acid sequence, and the same counts for untranslated regions. Even potentially less interesting, but yeah, it very much depends, of course, are intergenic variants, especially if you cannot associate them with a gene at all. So these coding variants, they can have different effects on the amino acid sequence. So as you know, a amino acid is translated from a triplet in the coding sequence. So if you have a mutation in the coding sequence, it can have an effect on the amino acid, but not always. So for example, if we are talking about a synonymous mutation, typically it doesn't have an effect at all at amino acid sequence. For example, ATA is translated into arginine, while ATG is translated into arginine as well. So probably this mutation, if the A is mutated into AG, probably doesn't have a big effect on phenotype. However, you can, of course, have a single point mutation, which can have an effect on the amino acid sequence. For example, in this case, when we have an AAT triplet that becomes an AAA triplet, so I think it's asparagine becomes a lysine, then of course the amino acid will change, and that can have an effect on the protein structure and therefore potentially on the phenotype. It becomes you can even get a larger effect when you gain a stop codon. For example, over here, this triplet would be translated TTT into a cysteine, but if that T mutates into an A, then it becomes a stop codon, and then you get a truncated protein. That means that the protein stops prematurely, and therefore probably it can have a very big effect on how the protein can do its job. Also, a big effect on an amino acid sequence are frame shifts. For example, if you have a single insertion or a deletion, then we get an out of frame translation. So you get completely different amino acid sequence resulting from the translation, typically also resulting in truncated protein. So these can also have a very big effect on the protein itself. So many things can happen, and it is always nice to be able to describe these different type of effects in a standardized way. And in order to do that, we typically use the sequence ontology that is a set of ontologies in a layered fashion that can help you or it helps software to describe particular variants. So it describes both the terms, so it also did definition and their relationships. So for example, if we have a coding sequence variant, it could be, for example, a protein altering variant, or a synonymous variant, or a terminated codon variant. And all of those different terms, they have a definition. And that definition we just discussed, right? If you think about proteins, it's also important to consider isoforms, because as you probably know, one gene can have different isoforms resulting into different proteins. It also means that one variant might be a protein altering variant in one isoform, but not in the other isoform, just because that exon is not part of that transcript. So how should we do that in terms of bioinformatics, right? Because we want to get a complete report, and if you and we want to find variants that have a big effect. So which isoform should we choose to report the variant effect? So there are multiple ways to do that. Typically, the effects of all isoforms are reported, at least all isoforms that have an exon in that region. But sometimes, if you are only interested in, a bit of an overview of what's possible, you can choose to only report, for example, the effect with the largest impact. So you focus on the isoform where the mutation had the largest impact on the amino acid sequence, or you focus on the most relevant transcript. And the most relevant transcripts are typically the transcripts or the isoforms that are most highly expressed. So there are definitely tools to do such functional annotation. Of course, in addition to a reference genome, you also need a functional annotation of that genome, meaning that you need to have the positions on where the genes and the exons and the introns are. Well, I give 3 examples. I think one of the most frequently used ones is VEP, Variant Effect Predictor from Ensemble. It both has a web interface where you can just upload a VCF and you get an annotated VCF back, or you can use the command line tool that interacts with the VEP API. Then there's ANNOVAR. Also relatively frequently used. I must admit I've never used it myself, but it is used by many people. And then there is SNPF. SNPF is a tool that we will also use in the exercises. The big advantage of SNPF is that it supports really a lot of different genomes, and the database is growing. And you can even make a database of your own genome, so your own annotated genome and GTF, and let SNPF make a database out of it, and then you can also use it for variant annotation. People have taken this a step further, because what people have noticed, and what you probably also noticed if you are doing a variant effect prediction, is that you can have a high impact mutation, for example, an amino acid change that might have a very big effect on the protein structure, but another amino acid change has only a minor effect on the protein structure, maybe because it's at the sites or at the inside of a protein 3D structure. So then people want to take that to a higher level, and what you can, for example, do is that you look for variants that are highly conserved in many species. So if you find a variant that is usually that changes amino acid that is highly conserved in many species, then you probably have found a variant with a relatively big effect, because it's probably important to keep it that way. Then you can also look in protein structure and biochemistry. You can imagine that AlphaFold might have had or has had a pretty big effect on that, because with AlphaFold you can exactly predict the effect one mutation has had on the protein structure. And also people have been using expression QTLs in order to see whether a certain variant has a big effect on expression of many genes. There are quite a few databases available. Examples of that is a DBNSFP and a mutation taster or taster, mutation taster.